Hello, dear students. Welcome back to GDC. In this class, we are talking about the different color reaction to identify a amino acid. The amino acids are basically combined to form a protein, and these amino acid need to be detected by the presence of different color reaction. So today we will discuss each and every method to identify different amino acid with different color, and we will solve some MCQ which will be going to be help you in your upcoming exam. Let's start. So these proteins are give color reaction against different chemical agent due to the presence of amino acids. So ultimately, this is a identification test for the protein, because the proteins are basically made from amino acid. If we will detect that particular amino acid, we can say this particular protein contains this type of amino acid. So these are the list of tests where we can identify different types of amino acids. This is a table. We will discuss each and every test in detail. First is biuret test. So biuret test is the most common test, and which is why it is used. It is used to check the peptide linkage, and at least two peptide linkage. That means at least three amino acid must be present over there. Okay. So next is mean hydrogen reaction. So this is for the detection of alpha amino acids. Xanthopoietic reaction. In xanthopoietic reaction, we basically detect the benzene ring. So basically the aromatic amino acid. So aromatic amino acid example is phenylalanine, tyrosine, and tryptophan. Okay, phenylalanine, tyrosine, and tryptophan. Next is Million's reaction. This is for the phenolic group present in tyrosine. Hopkins-Cole reaction, detection of indole ring. Sakaguchi reaction, detection of guanidino group present in arginine. Nitroprusside reaction and sulfur test for the detection of cysteine, sulfide group present in cysteine. Pauli's test for the imidazole ring present in histidine. Polycyclocalcium test for the phenolic group present in tyrosine. So these are the what we can call, these are the just overview. I just show you in a tabular form so that you just understand what we are going to discuss about. Let's start discussing about each and every point. So first test is biuret test. So biuret test, why it is used? It is used for the peptide linkage. Now one question always asked about how these peptide linkages are formed. Let me explain you how a peptide linkage is formed. This is also one other question. Peptide linkage. So let me give an example. For suppose this is one amino acid. Okay, this is one amino acid, and this is another amino acid. R1CH NH2C4. What happens here basically? So this carboxylic group and this amino group interact with each other. Okay, this carboxylic group and this amino group interact with each other, and there will be elimination of a water molecule and formation of your what? Your peptide bond. So this is your peptide bond. Okay. So question will be asked. So how these peptide bonds are formed? Peptide bonds are formed from the carboxylic group from one amino acid and amino group from another amino acid. When fused, there will be elimination of a water molecule and there is a formation of this peptide linkage. So in this biuret test, what we basically detect? We basically detect the peptide linkage and at least two peptide linkage. That means three amino acids should be joined together. Okay, so this is gives a positive test. So by the way, it is specially detect the peptide linkage and detect the protein in the biological sample. For suppose you want to check the protein level in urine, or suppose you want to check the protein level in the blood. In such cases, also this biuret test can be used. So how this biuret test basically works? So when urea is heated over 180 degrees centigrade, so biuret is formed. And this biuret basically treated with dilute copper sulfate in an alkaline medium to give a purple color. Okay, that basically detect the presence of protein or peptide. Please note it down. What happened? First, you have to heat the urea over 180 degrees centigrade. It will generate biuret. Now, this biuret react with dilute sulfuric acid dilute copper sulfate in an alkaline medium, it will develop a purple color, and that will give basically detect the presence of protein or peptide. So that is basically bioreactor. This is used for the detection of the peptide linkage in a biological sample. Okay, next test. So next test is, I hope you understood this, what is this bioreactor test and what is its principle. Next test is mean hydrogen test. So mean hydrogen test is very important. Many times this question have asked. So mean hydrogen test is why it is used, is basically used for the detection of alpha amino acid. Please note it down. It is used for the detection of what? Alpha amino acid. So when alpha amino acid react with nein hydrogen, this is a reagent called nein hydrogen, and it will give to this nein hydrogen, there is a development of purple, blue, or pink color. So the color that we develop, we call it as a Riemann purple. So many times this color is about asked in exam. Please remember Riemann purple. What is the color? That is Riemann purple. Okay. So if we talk about this methodology, when amino acid react with a nein hydrogen, it will develop a ketoacid along with ammonia, carbon dioxide, and nein hydrogen. So first is amino acid, first nein hydrogen, it develop a ketoacid, ammonia, carbon dioxide, and hydrantine. This hydrantine again react with ammonia and nein hydrogen will result in Riemann purple color. This is the important point. Okay, which color it is developed, because this color reaction you can directly see. I will show you. I will practice some of the question. You will understand what type of question they basically ask. So they basically, you will see the question like uh, white color precipitate is developed in which test, in nein hydrogen reagent and nein hydrogen reaction. What are, which type of amino acid we detect? What is the color that develop in nein hydrogen reaction? So this type of question you might see in your in the exam. So such type of question can be asked in exam. So please note it down. In case of nein hydrogen test, the humans purple is developed. Okay. Next question. Next question is, next method is xanthopoietic test. What this xanthopoietic test basically means, let's talk about. So this xanthopoietic test, what it do, it basically detect the aromatic amino acid. Aromatic amino acid. So this aromatic amino acid, if we'll talk about, so there are three major type of aromatic amino acid are present. So one is phenylalanine. Second one is tyrosine. Third one is tryptophan. Please note it down. Three amino acid, aromatic amino acid group, that is aromatic amino acid present in the structure. And this xanthopoietic test, what it is basically used, it is basically used for the detection of this phenylalanine, tyrosine, and tryptophan. So these aromatic amino acids react with strong HNO3 nitric acid at high temperature. There will be a nitration of this reaction and result in color. And this basically called as phenyl. This is basically detection of aromatic amino acid. Okay. Now I'm moving to the next test. That is what we can call millions test. So what is this millions test? Million test, it gives a positive test for tyrosine. Okay. Or hydroxyphenyl. Actually, what million test basically do? So million test basically detect a aromatic ring with a hydroxyl group. That is, we call it as a hydroxyphenyl group. So this, if a amino acid having this type of structure, it will detected by millions test. So millions test is also used for the detection of tyrosine as well. So this is basically detect what a structure should have to be detected by millions test. You should understand that is your amino acid with hydroxyphenyl ring. What is that? Amino acid with hydroxy. If a amino acid have hydroxyphenyl ring, then it can be detected. Understood? I hope you guys are understood. It should have this group. This group, its name is hydroxyphenyl group. What is the name of that group? Hydroxyphenyl group. Okay. Next is Hopkins-Cole reaction. What is Hopkins-Cole reaction? So this is for the amino acid having indole ring. So tell me which amino acid have indole ring? Comment down your answer in the comment section. Which amino acid have indole ring? So when we heard about this indole ring, we always remember the amino acid that is your tryptophan. What is the name of that amino acid? The name of that amino acid is tryptophan. So tryptophan basically causes which ring? It causes indole ring. Which ring it should have? It should have indole ring. So this indole ring containing amino acid, that is tryptophan, react with formaldehyde, sulfuric acid, and mercury sulfate. What happens? Four reagent. First one is uh, formaldehyde, sulfuric acid, and mercury sulfate. It will develop a violet color or purple color complex. So for why this is used? So the color that develop is violet or purple color. So why it is used? It is used for the detection of indole group present in amino acid. Which amino acid have indole ring? That is tryptophan ring. When tryptophan react with formaldehyde, sulfuric acid, and mercury sulfate, it will lead to a violet to purple color, and that is basically detecting what? That basically detects the that basically detect what? That basically detect the tryptophan amino acid having indole ring. This is a simple fundamental concept. I'm just trying to making your concept clear. So first thing is we discussed about biorate test. So biorate test, we check that this is about the detection of peptide linkage. Next we talked about mean hydrogen test, where we detect where we see that this is for the detection of alpha amino acid. Third, we see the xanthopoietic test for this aromatic amino acid. Next, we see about millions test. That millions test is for the detection of phenyl hydroxyl group. Next, we'll check about Hopkins-Cole reaction. This is for the amino acid having indole ring. That is for the tryptophan. Next is Sakaguchi test. So Sakaguchi test, why it is used? It is used for the arginine detection due to presence of guanidino group. The Sakaguchi test basically detect the guanidino group. So this guanidino group basically present in which amino acid? That is arginine. It is present in arginine. When this arginine react with alpha naphthol in the presence of alkali hypobromide, it will give a red color. Which color it will give? It will give a red color. Please note it down. So first is your Sakaguchi test. So Sakaguchi test is for the detection of guanidino group. Guanidino group present in which amino acid? That is arginine. When this arginine react with alpha naphthol in the presence of alkali hypobromide, you will find a development of red color. And due to this development of red color, it will detect the arginine. So arginine have which group? Guanidino group. Next test is sulfur test. So sulfur test, this is the only give test positive for the sulfur containing amino acid. Yes, tell me what are the name of sulfur containing amino acid. Comment down your answer. Which amino acid should have sulfur atom in its structure? Yes. Which amino acid have sulfur atom in its structure? There are three amino acids. What are these three amino acids? One is your cysteine, another one is cysteine, and third one is for methionine. So this sulfur test is for the detection of two amino acids, that is cysteine as well as cysteine, not for the methionine, not for the methionine, because the ring the what there is a concept why it is not give a positive test for the methionine, because methionine is never hydrolyzed in the presence of alkali. So due to that reason, methionine does not give positive test for the sulfur test. So when it react with except methionine, because methionine does not give a positive test. Amino acid when boiled with NaOH alkali medium, organic sulfur is converted to inorganic sodium sulfide. Now the sodium sulfide is react with lead acetate to produce the lead sulfide, that is black precipitate. Question asked, which color reaction produce a black colored precipitate, that is sulfur test. I will explain you with a mnemonic so that you can remember the what are the tests and what is its application. Again, I am repeating that test. So what happens? So this sulfur test, this is for the detection of what? Sulfur containing amino acid. So there are two sulfur containing amino acid. One is your cysteine, another one is your methionine. So cysteine and cysteine. Cysteine and cysteine are both are having sulfur atom. Okay. So next is methionine. So methionine does not show this reaction positive because methionine cannot hydrolyze in the presence of alkali. And there is a development of black color. What actually test is when amino acids react with boiled alkali, it convert organic sulfur into inorganic sodium sulfide. Now, sodium sulfide, when react with lead acetate, it forms lead sulfide, and that lead sulfide basically produces a black color precipitate. So that is basically for the detection of what? That is basically for the detection of your uh, amino acid cysteine and cysteine. Next is polystate. So polystate, this is specifically for the imidazole containing ring. So this is for the imidazole containing ring. Now question will be asked, which amino acid contains imidazole? So imidazole containing amino acid is your histidine. When diazotized sulfonic acid react with imidazole ring, in case of alkaline condition, you will develop a red color, and that red color is for the detection of what? That is for the detection of your histidine amino acid. Which amino acid? Histidine amino acid. And specific detection on Mollis test. So Mollis test is for the detection of glycoprotein. Okay, Mollis test is used for the detection of what? Glycoprotein. I hope you guys are understood what are the different tests and what is basically application of the test. Now, I will summarize what are the things that we have learned. Okay, we will summarize in a tabular form. Okay, so first the mnemonic is big naughty X-Men makes heroes save super people's molecule. So B stands for biuret test. Biuret test is for the peptide bond linkage and it develops purple color. Nein hydrin test for the alpha amino acid, it basically developed a Reuman's purple color. Xanthoprotic test is for basically aromatic amino acid gives yellow to orange color. Millions test is for the detection of tyrosine, it gives a red color precipitate. Hopkins-Cole reaction for the tryptophan amino acid, it gives a violet color. Sakaguchi test for the arginine amino acid gives a red color. Sulfur test, cysteine and cysteine, black color precipitate. Paulis test, cysteine, red color. Mollis test for a purple ring. Okay, so you see, this is the test detects what and its color reagent. So biuret test detect peptide bond. Nein hydrin test detect alpha amino acid. Xanthoprotic test detect aromatic amino acid like tyrosine, tryptophan, and phenylalanine. Millions test for the tyrosine detection. Hopkins-Cole reaction for tryptophan, Sakaguchi test for arginine, sulfur test for the sulfur-containing amino acid except methionine, Paulis test for the histidine imidazole ring, okay, Mollis test for the carbohydrate glycoprotein. So these are, I think, you guys are understood what we have discussed right now. So we discussed the test, we discussed what this test basically detect and its development of color, which color it is exactly developed. I hope there is no confusion till this point. So if there is no confusion till this point, then let's discuss some of the MCQ so that your concept would be more clear. And now when I start discussing MCQ, you might have to give the answer simultaneously and I will give 20 to 30 seconds to think and answer. Comment down your answer. I will check how many of you are given the correct answer. Let's start. So first question. The biuret test specifically detects. So this is basically this question was asked in Gujarat DI 2021. The biuret test basically detects what? Alpha aromatic, alpha amino group, aromatic ring, peptide ring, yeah, indole ring. Yes, tell me. The biuret test specifically detects what? Alpha amino acid, aromatic ring, peptide bonds, yeah, indole ring. Comment down your answer in the chat box as well as in the comment section so that I could check and I will analyze how many of you have given the correct answer. Yes, which one is the correct answer? The correct answer is peptide bond or peptide linkage. So let's see. So alpha amino group is detected by, yes, ninhydrin. Okay. Aromatic ring is detected by, yes, xanthopoietic test. Indole ring is detected by which test? Yes, Hopkins-Cole reaction. This is present in tryptophan and this is detected by Hopkins-Cole reaction. Okay, always check which option is correct and which other option is not correct and why it is not correct. That will analyze the things in a better way. Now moving to the next question. Formation of Riemann purple color. Formation of Riemann purple color is basically used, is observed in which test? Yes, tell me. Formation of Riemann purple color is used for that is observed in which test? Mean hydrin test, xanthopoietic test, millions test, या Pauli's test. Tell me. Easy question. Easy question. This type of question asked in exam. Okay. So that's why I kept such type of question for your better understanding. Formation of Riemann purple is observed in which test? Mean hydrin test, xanthopoietic test, millions test, या Pauli's test. Which test is it basically? The correct answer is yes, mean hydrin test, na? In case of mean hydrin test, I told you, na, Riemann purple color is developed. Let again see the table, you will understand. So in case of biuret test, you will give a violet to purple color for due to peptide linkage. In case of mean hydrin test, you get a Riemann purple color due to this alpha amino acid. You have xanthopoietic test for the aromatic amino group. It basically detects the orange color, yellow to orange color. Millions test, that is for the tyrosine amino acid, it basically detects if they develop a red precipitate. Hopkins-Cole reaction for the tryptophan amino acid, it also gives violet or purple color. Sakaguchi test, it will give a red color. Sulfur test, it will give a black color precipitate. Pauli's test, it will give a red color. And Molly's test basically gives a purple ring at its interference. Okay? Okay, I hope you understood. Next question. Which amino acid give a positive test Hopkins-Cole reaction? Just now we will discuss. Just now we have discussed Hopkins-Cole reaction. Why it is used? Answer this question. Why this Hopkins-Cole reaction is basically used? For the detection of tyrosine, for the detection of histidine, for the detection of tryptophan, or for the detection of arginine. The correct answer is tryptophan. Okay, so Hopkins-Cole reaction basically detects the tryptophan. Tryptophan contains which amine ring? Indole ring. Tyrosine is detected by which test? Phenyl tyrosine is which type of molecule it is? It is basically a aromatic ring. So aromatic rings means it can detected by xanthopoietic test. Now histidine imidazole ring. Histidine imidazole ring detected by which test? Who can tell? Histidine imidazole ring. See, Pauli's test. If it will have imidazole ring, then it will detected by this your Pauli's test. Okay, this type of question, this table. If you remember this table, na, if you will not remember the chemical name and all, but if you remember this table only, it will more than sufficient to answer all the questions. So always you remember this table, keep this table in your mind. Okay. So here the correct answer is tryptophan. Now moving to the next question. A protein solution turns red when treated with alpha naphthol and alkaline hypobromide. Which test it is? Yes, very easy question. A protein solution turns red when it reacts with alpha naphthol and alkaline hypobromide. Which amino acid it conforms? Please remember in which cases we see this alpha naphthol and alkali hypobromide is react and red color last test. Yes, remember which test first you identify which test name is. Then you will easily answer the amino acid. Recall what we have learned. Come to this slide. What we have learned here. When this guanidino group react with alkali hypobromide, come to this slide. When this arginine group having guanidino group, okay, react with alpha naphthol and alkali hypobromide, what it will develop? It will develop a red color. This is for the detection of which amino acid? Yes. What is the correct answer? Which amino acid it is detecting? It is detecting arginine. Okay, it is detecting arginine. So I want to tell you that you also remember the sum of the name. That is, it is for the arginine. Cysteine is detected by which test? Sulfur test. Okay. Histidine, Pauli's test. Phenylalanine, xanthoproetic test. Okay. Next question. Sakaguchi test. Okay. Next question. Black color precipitate in sulfur test is indicate the presence of black color precipitate in the sulfur test. It is indicate the presence of methionine, cysteine, phenylalanine, yeah, histidine. Yes. Very easy question. Very direct question. You all of you can easily answer. Yes. Give me the correct answer. Comment down your answer in the chat box as well as in the comment section so that I will check how many of you have given the correct answer, and if any wrong answer is there or if anything you also want me to ask also, you can write your question in the comment section. I will give you the correct answer for your question. So black color precipitate. So black color precipitate means it is for the detection of sulfur test. So sulfur test, what is the rule I told you? I told you, you remember three sulfur containing amino acids. One is your cysteine, one is your cysteine, one is your methionine. So methionine does not give a test for the sulfur test, but the cysteine and cysteine will give for positive for the sulfur test. Option B is the correct answer. Okay. Next question. Xanthoproetic test is used to identify sulfur containing amino acid, aromatic amino acid, histidine, या alpha amino acid. Yes. Why this xanthopoietic test is used? Why this xanthopoietic test is basically used? So first you remember what we do in xanthopoietic test. So xanthopoietic test is basically detect what? It basically detecting aromatic amino acid. Aromatic amino acids. What are the example of aromatic amino acid over here? One is your phenylalanine. Second one is your tyrosine. Third one is your tryptophan. Okay, there are three amino acids which is basically detected. So option B is the correct answer here. Okay, now moving to the next question. Pauli's test. Pauli's test is for the detection of tyrosine, histidine, tryptophan, या arginine. Simple question, very direct question. So you would give the answer. Pauli's test is specific for the detection of tyrosine, histidine, tryptophan, and arginine. First, you remember Pauli's test detect which heterocyclic ring? Pauli's test detect which heterocyclic ring? Remember, Pauli's test detect which heterocyclic ring? Can I say it detect this ring? Can I can I tell this idea polyester detect this ring, whether it is correct or not? Tell me. Imidazole ring. Very good, na? Imidazole ring detect. So imidazole ring present in which amino acid? Histidine amino acid. So the correct answer will be option B, histidine. Okay. Next question. A carbohydrate moiety in glycoprotein is confirmed by Mollis test, Biuret test, Xanthoprotic test, or Sakaguchi test. Answer this question. A carbohydrate moiety in glycoprotein. So glycoprotein means glycogen along with protein. That is sugar along with protein. So the sugar moiety in that protein is confirmed by whether it is by Mollis test or Biuret test or Xanthoprotic test, yes, Sakaguchi test. Yes. Answer this question. Which one is the correct answer according to you? Carbohydrate moiety. So when you always focus with the terminology. Carbohydrates. So when we talk about carbohydrate moiety, so Mollis test is the one. So Mollis test is basically detect the glycoprotein. Okay. Mollis test basically detect the glycoprotein. Okay. Next question. Alpha amino acid react with nein hydrine. Alpha amino acid react with nein hydrine to give a color complex of yellow, pink, black, yeah, purple. This question was asked in Bihar DI 2008. So alpha amino acid react with nein hydrine to give which color? Yes, alpha amino acid react with nein hydrine to give which color complex? Whether it is a yellow, whether it is a pink, whether it is a black, or whether it is a purple. Why this alpha amino acid is basically reacts? So this alpha amino acid react with nein hydrine test to give which color? To give purple color. Okay. Nein hydrine test for the detection of alpha amino acids. Okay. Last question of this today's class, that is an amino acid gives a yellow precipitate in the xanthoprotic test, and it was asked in UPSC DI 2019. An amino acid that gives a yellow color precipitate in case of xanthoprotic test. So first question, why xanthoprotic test is used? This is for the aromatic amino acids. So what are the example of aromatic amino acid? Phenylalanine, tyrosine, and tryptophan. So here tyrosine, option C. So, okay, option C is the correct answer. So I hope you guys are understood what we have discussed today. So we basically discussed in this class basically about the color reaction of amino acid. So we check here what are the different type of color reaction for the different detection of different type of amino acid. We see that based on the different heterocyclic based on the different structure, this color reaction perform, and we basically detect the specific amino acid. I hope you guys are understood, and if you have any question or any query, you can comment down in the comment section. So stay connected with the GDC for more such important content. Thank you.